We have reached the top of the hour, so let's begin.
Welcome, everyone, and thank you for joining today's webinar, Optimizing Poultry Gut Health from Microbial Foundations to Disease Management. On behalf of the Animal Nutrition Association of Canada and the Poultry Science Association, we're excited to bring you another installment in PSA ongoing webinar series, designed to highlight timely research and practical insights across the poultry sciences. I'm Jocelyn Zafra Castillo from the Animal Nutrition Association of Canada, ANAC. ANAC is the Livestock Feed Industry National Trade Association. Our 189 members include mixed feed and ingredient manufacturers and distributors, as well as suppliers of a wide range of goods and services to the feed industry. Taken together, ANAC's membership represents 90% of commercial feed manufactured in Canada. ANAC proudly represents the Canadian livestock feed industry in advocating with policymakers for a fair regulatory environment, collaborating with industry partners, and supporting members in their day-to-day business. And now I'm handing it over to Lindsay to introduce the PSA.
Thank you. All right. So the Poultry Science Association is a global professional org that consists of approximately 2,000 educators, scientists, extension specialists, industry researchers, administrators, producers, and college students who are committed to advancing the poultry industry. Since 1908, PSA has maintained a level of prestige that ranks it among the top professional organizations in the field. And we own three academic journals: Poultry Science, the Journal of Applied Poultry Science, and European Poultry Science, which we all hope you'll look into.
So now jumping into today's session. Today's session will explore the vital role of the gut microbiome in poultry health, from early microbial development to managing disease challenges like Eimeria. Gut health is central to maximizing productivity, supporting nutrient absorption, and reducing reliance on antibiotics in commercial production systems. The information shared today is highly relevant for producers, veterinarians, researchers, and students alike. We're honored to be joined by two outstanding speakers today, Dr. Ivan Rychlik from the Veterinary Research Institute in Brno, Czech Republic, and Dr. Philip Yeboah from the Prestige Department of Poultry Science at North Carolina State University. But before we dive in, just a quick reminder, the Animal Nutrition Conference of Canada and the PSA annual meeting are right around the corner. ANAC's Animal Nutrition Conference will take place from May 5 to 7 in Edmonton, Alberta, and the Poultry Science Association annual meeting will take place in July 10 to 13 in Toronto. So you can scan the QR codes or access the website links that we will be sharing in the chat for more event details and registration information. Some Zoom housekeeping for our webinar today. The webinar is being recorded and will be available on the Poultry Science Association website within 36 hours of today's conclusion. If you have a question, please submit it using the Q&A tab by clicking the Q&A icon at the bottom of your screen. Please do not use the raise hand feature. Attendees are not permitted to speak during the webinar. When you submit a question in the Q&A tab, it may be answered in writing or flagged to be addressed live by our speakers, and we will reserve time at the end of the webinar for a live Q&A. You're welcome to like or comment on other attendees' questions. Questions with more likes will be prioritized during the Q&A session, so please like questions you'd also like to see addressed. If time does not allow us to answer every question, we will move through them based on priority. The chat function is disabled, but again, please feel free to submit questions through the Q&A tab. Also, live captions are enabled for today's webinar. If you prefer not to use them, you can turn them off by clicking Hide Captions at the bottom of your screen. So now moving on, it's my pleasure to officially introduce today's speakers. We have Dr. Ivan Rychlik, is a professor of microbiology and a group leader at the Veterinary Research Institute in Brno, Czech Republic, where he studies the chicken gut microbiota to improve gut health and production outcomes. His work led to the development of QuoCNA, a defined probiotic now commercially available for use in newly hatched chicks. And Dr. Philip Yeboah is a postdoctoral research fellow at North Carolina State University, whose research focuses on poultry gut health, microbiome dynamics, and disease mitigation. He has worked extensively with major poultry pathogens and developed innovative in vitro and in vivo assays to evaluate vaccine efficacy and microbial viability. Thank you everyone for joining us today, and thank you to our wonderful speakers. We're so excited to have you. And with that, I'm turning over things to our first speaker, Dr. Ivan Rychlik.
Hello, everybody. Transatlantic hello from the Central Europe. And I will now share my screen and start my presentation. Fine. First of all, I would like to thank Jocelyn and Lindsay for inviting me to this webinar, to this online conference meeting. And since 15-minute slot is quite a short one, I will immediately move to my presentation, to its content. So all of us, we know that chickens in commercial production are hatched in hatcheries, and from hatcheries are moved to farms in where they are kept in tens, thousands animal flocks. And all of us, because of daily routine and numerical dominance, we consider this as a normal, as a reference, as a somehow standard. However, despite this numerical dominance, this is not correct assumption. Normal is something to which biological species, Gallus, evolved throughout the whole millions years of evolution. And normal means for chicks to be hatched in a nest and to remain from the very first moment of their life in a contact with parents, with adult hens, parents, and rest of the flock. This difference is pretty important for the development of chicken gut microbiota. We perform multiple experiments in which we transfer fecal material, cecal contents, different extracts, and then we ended up with the simplest experiment in which we kept chicks either on their own, mimicking commercial conditions, so chicks from the hatcheries, and then to another group of chicks, we added an adult hen. This was not the mother of these chicks. This was really an average standard egg-laying hen from the middle, which we collected in the middle of lay. We placed them in physical contact, and when these chicks were either 6-day-old or 12-day-old in both groups, those without any contact and those in a contact with an adult hen, we determined by DNA sequencing, 16S rRNA sequencing, we determined composition of cecal microbiota. And at the very first immediate impression, you can see that the cecal composition of microbiota composition differs in chicks which were kept in alone on their own, and those which were kept in a contact with an adult hen. Those kept alone, their microbiota dominated by gram-positive bacteria belonging to phylum Firmicutes. However, those which were kept in contact with an adult hen, their microbiota was complete or could not be differentiated from the microbiota of the donor hen. So a new group belonging to phylum Bacteroidetes, this new group of bacteria appeared in cecal microbiota of the chicks which were in a contact with an adult hen. And it is also well known that these chicks from the hatcheries, are nicely or heavily pretty sensitive to different infections of intestinal tract, while those chicks which are inoculated by fecal material or by contact with an adult hen, these are nicely resistant, for example, to Salmonella challenge. So we started to be interested in these bacteria, but this figure also tells you a couple of additional details or information. What is, for example, the origin of these bacteria? Well, environment. There is no donor hen. So all these bacteria are not chicken specific, not at all. Those are bacteria of environmental origin, which are also capable of replication in chicken intestinal tract. This experiment and data also completely denies the dogma on the age development of chicken gut microbiota. This is only a consequence of the fact that we are always handling with chicks from hatcheries. But there is no age-dependent development. It's only a question of lack of the resources. If you add a proper source, parent, adult bird, contact, chicks can be colonized by adult type of microbiota from the very first days of their life. On day 6, their microbiota was completely of the adult type. You can also deny or exclude the hypothesis on necessity of anaerobization of chicken intestinal tract. This is not true. From the very first moment, from the very first days of life, chicks can be colonized by any bacteria which are appropriate and suitable for chicken intestinal tract. So we, in our research activities, we then focused mainly on these bacteria belonging to Bacteroidetes. We performed multiple strictly anaerobic cultures, picked up thousands of colonies, and now I skip 10 years of our research. We ended up with a product which consists of 9 strictly anaerobic bacteria. 6 of them belongs to phylum Bacteroidetes. 3 of them, 2 Megamonas species and Megasphaera, belong to phylum Firmicutes. And when these bacteria, this mixture, is administered to chicks on day 1 of their life, just after hatching, and if these chicks are on day 7 challenged with Salmonella, so those chicks which were treated, experimental ones, were and are as well more resistant to Salmonella challenge, and the difference is somewhere around 2 logs of magnitude. So chicks colonized by that type of mixture are 100 times more resistant to Salmonella challenge. Having this mixture, we passed through the approval process, and since 2024, this is in Czech, but doesn't matter. Since March 2024, we are allowed to distribute this mixture in the Czech Republic only to newly hatched chicks to increase their resistance to Salmonella infection. And we somehow, and we do have some evidence, extrapolate that if more resistant to Salmonella challenge and Salmonella infection, that such chicks might be more resistant to other enteric diseases as well, which is the case. Last year, in 2025, when we started the full commercial distribution, we treated approximately 5% of chicks produced, mostly broilers, chicks produced in the Czech Republic with this mixture. Farmers and users were quite satisfied, and this year our sales and distribution success, I may perhaps even say, is still increasing. So it looks that we will cover more than 15% of the market, Czech market, with the Czech broiler market with this product. In all the cases when these bacteria, this mixture is administered, farmers do not use antibiotics. So besides increasing resistance to Salmonella, we also decrease antibiotic consumption in the Czech Republic. During the commercial distribution, we of course received plenty of different feedback comments from the users, and one of them, both feedback and our way of thinking, our way of further thinking, we always, when we tested this mixture experimentally, we always administered this mixture to 1-day-old chicks, newly hatched chicks, and we waited for 1 week and let these bacteria somehow to establish in the chicken intestinal tract. 1 week, it is fine, but most of the mortalities and fatalities and losses in the chicken production, in broiler production, appears and happens during the first 3 or 4 days of life. So waiting till day 7 from a farmer point of view, if you do not think for Salmonella only, but if we were thinking from a farmer point of view, losses, mortalities, day 7 might be too long, too late. So we reconsidered, returned back, and we started to be interested, and I will be talking more about this during the animal nutrition conference in Mountain, more details I will show, but we started to be interested in microbiota composition in that mistakenly or miscorrectly developed chicken embryos during the incubation. We start to be interested in the microbiota in this material, eggshell waste, when the hatching in hatcheries is accomplished. We then looked at cecal microbiota of chicks just during the hatching process. We looked and analyzed microbiota, cecal microbiota of chicks immediately after placement on farms, but before they started to consume feed. And then we also looked at microbiota, cecal microbiota of 2 days, 4 days, 6 days, and 7 days, 7-day-old chicks. And details and characteristics, all the composition I'm not showing, but what we observed, two bacterial species, 2 different bacteria, Enterococcus faecalis and E. coli. These were the only 2 bacteria which were present in all these different samples. Sometimes they were found in corrupted chicken embryo. These were common in eggshell waste. These were common also in 1-day-old and 2-day-old chicks. And E. coli continues to expand and proliferate in chicken cecum till the age of day 7. Fine. Now, I will focus only on day 1 chicks, day 1-day-old chicks. Whenever we analyzed, the presence of Enterococcus faecalis or E. coli was, I would say, digital, black and white, 0 or 1. Either analyzed chicks were completely free of Enterococcus faecalis or E. coli, or when these bacteria were present, they formed nearly 100%. So this was the only bacterial species which was present in there. And this is the same for individual chicks, and the same story for Enterococcus faecalis or E. coli, present or highly dominant, numerically dominant, completely dominating. And because of these, although I am generally critical towards the use of bacteriophages, we started to consider that if in the intestinal tract of 1-day-old chicks, just at the moment of placement on farms, if there are nearly monocultures of Enterococcus faecalis or E. coli, maybe in that particular case, it might be possible to use phages. So we started to purify phages because the interaction is quite likely. There is nothing else. So when you administer the phages, bacteria target is in there, and there is no interference with other bacteria, with other microbiota. So we started to purify phages, and it is very common and very simple to purify and extract and obtain phages from eggshell waste or from the litter when the flock is approximately a few days old. We usually visit farms on day 4. So in that material, in the litter or in the eggshell waste, it is very simple to find E. coli phages and Enterococcus faecalis phages. Surprisingly, it is pretty difficult, for example, to find bacteriophages against Proteus, Enterococcus faecium, and Klebsiella, Enterobacter, or anything else. So the fact that bacteriophages against Enterococcus faecalis and E. coli can be found in this material also somehow confirms the fact that this type of material in these days is really dominating, can be dominated by these bacteria. So we start to test them. Initial experiments are of a moderate success, but it seems that if we combine the phages together with our probiotic product, which I introduced in my previous slide, that these may contribute and increase in a synergistic way and increase the efficacy of the treatment. Fine. And then what we also do, what we also analyze is microbiota. Oh, sorry. Microbiota in the actual waste. Why? This is quite clear, because chicks immediately during the hatching, when these are being hatched, are immediately exposed to bacteria which can be present in that type of material. And so we started to analyze again by DNA sequencing, 16S RNA sequencing. And that's what we noticed, that out of 60 different samples which we analyzed, we now analyze many more, but when we started, so this is one of the earlier figures, some 5 of them were positive, or in these samples we also find DNA from Salmonella. So these samples were positive for Salmonella, approximately 10%. Whenever Salmonella was present, we also noticed that these 2 bacteria, Chrysobacterium and Sphingobacterium, were absent. However, when these 2 bacteria were present in the actual waste, Salmonella was always absent. So we started to be interested in these 2 bacteria for the potential anti-effects against Salmonella in hatcheries, in that type of material. And in addition to self-correlation of microbiota in this type of sample, since we were in a good collaboration with the hatchery personnel and also with the farmers, we received and obtained data on the performance of these chicks when placed on farm. So we could correlate microbial composition in that type of material with mortalities reported by farmers 1 week, 2 weeks, 3 weeks, and 4 weeks after placement of chicks at farms. Correlation was quite weak, but we could observe that when different actual samples were dominated by Enterobacter, Pseudomonas, Enterococcus, or Klebsiella, farmers usually reported higher losses during the first days after chicken placement on farms. On the other hand, although the correlation coefficient was quite low, 0.24 only, when Chrysobacterium and Sphingobacterium dominated in actual waste, farmers usually reported lower problems with the chickens, lower mortalities, and so on. And when we combine pretty low or rather low, quite small, but positive correlation, together with anti-Salmonella effects, we started to use mixture of these 2 bacteria to protect environment of the hatcheries. Chicks or chickens are not the direct target of the treatment. The chicks are indirect consequence of the treatment, but we first of all treat the surface, floors, walls, technology, all the technology, and in that type of environment, we suppress or reduce the multiplication of unwanted microbiota members. So if I am to summarize, what we are running, and this is only because of the 15-minute slot, right? What type of experiments we are running these days? We use our research results of our research, and we spray mixture of Sphingobacterium and Chrysobacterium on day 20 of embryonic development, so -1 day in terms of hatching of the chicks. And target is in the hatcheries. Target is hatchery environment. Newly hatched chicks on day 1 or 0, it depends how you want to view it, are now treated with our product, Quochna, but in fact you can use whatever alternative probiotics or competitive exclusion products. And then when the chicks are placed on the farm, we supply via drinking water phages mainly against E. coli and Enterococcus faecalis, usually for the first 3 days of fattening. We also consider to treat eggs, not for human consumption, but for broiler production, after disinfection with some microbiota, because to disinfect and let the empty ecological niche of the egg open and available to any microbiota from an environment, it's quite risky. So we are considering that Chrysobacterium and Sphingobacterium, maybe in a combination with lactobacilli or maybe in a combination with some specific phages, can be used also for the treatment of surface of the eggs. But this, at the moment, I think we tested this only once, which means 0, and we do not develop this idea at the moment. Whenever we combine all these 3 steps, chickens are then produced without any use of antibiotics. So we increase the resistance to enteric diseases, and we also decrease antibiotic consumption. And in that way, we protect not only the chickens, but also human population. Thank you for attention.
Thank you so much, Dr. Rychlik, for this incredibly insightful presentation. And as he mentioned, if you want to learn more about this, please join us for the Animal Nutrition Conference. And now I'm going to hand it over to Dr. Philip Yeboah for another wonderful presentation.
All right, thank you so much, Joyce, for the introduction, and also thank you, Lindsay, for the invitation. So I want to present on the topic, metabolic disruption of the microbiome during Eimeria infection. Then I'll focus my discussion some technical. Okay. Right, I'll focus my discussion on the shift in the microbiome composition and metabolism during Eimeria infection. I will talk about some potential biomarkers for disease monitoring, then also to highlight some insight for targeted gut health interventions. Now, as we may be all aware, Eimeria is a huge threat to the poultry industry, with estimated global annual loss of $14.4 billion. In the US alone, it costs the chicken industry $1.6 billion a year. Now, vaccination remains a very sustainable tool for controlling Eimeria. However, vaccination is a controlled infection. Now, during peak cycling, the epithelial cells are damaged, then inflammation increases, then the gut environment also changes. This throws the gut microbiome into an imbalanced state, which eventually lead to temporary reduction in feed intake and also temporary reduction in body weight. Now, what is gut health? If we talk about gut health, a gut is said to be healthy when it's able to operate without major form of stresses. It is able to maintain homeostasis, and it is able to perform physiological functions without any major hindrance. When this happens, birds are able to achieve close to 100% of their genetic potential. Now, a healthy, stable gut microbiome providing functions such as host synergy, protected mucous membrane, colonization resistance against pathogenic bacteria, balanced immune activation, and proper nutrient utilization is the golden standard we all hope for. However, this is not always the case because birds are challenged with multiple stresses, which leads to dysbiosis, thereby leading to damaged epithelium, increase in toxin buildup, reduction in diversity, unbalanced immune response, increase in cellular turnover, and improper utilization of nutrients. All these put together negatively impacts the performance of birds. Now, Eimeria infection is a major part of these stresses. When Eimeria enters the gut, it replicates within the gut. Then it leads to the leakage of nutrients into the gut lumen, and opportunistic bacteria utilize this nutrient to proliferate, thereby throwing the gut microbiome into an unstable form. Now, existing approach in many vaccination programs deals with the use of supplementation of feed additives such as probiotics, phytogenics, essential oils, organic acids. But these are very great tools to use. But the question we ask ourselves is, how precise are we selecting these feed additives? Are we selecting these feed additives based on what is happening in the gut at specific time points when we vaccinate, or we are using them as a broad spectrum control method? We believe the gut, Eimeria induce very specific changes in the gut. Therefore, our interventions must also be specific and targeted. Therefore, we ask ourselves a very simple question. How does vaccination impact the gut microbiome of turkey poult at critical time point from day 4, day 9 to day 14 when we vaccinate? We believe we are able to understand what happens, the dynamics in the gut microbiome. We can also select targeted interventions to support birds during vaccination. Therefore, we set a very simple hypothesis. And our hypothesis was multi-species Eimeria vaccine alters cecal microbiota of turkey poult by decreasing alpha diversity, shifting beta diversity, enriching harmful bacteria, depleting beneficial bacteria, and downregulating essential metabolic pathways. And our objective was to evaluate how multi-species Eimeria vaccine affect alpha and beta diversity, harmful and beneficial bacteria, and also essential metabolic pathways regulations in the cecal microbiota of turkey poult. So for our materials and method, day of hatch turkey poult were subjected to 2 treatments. We have the control and the vaccinated group. Each treatment contained 20 poults. Then on day of hatch, those in the vaccinated group were inoculated with 1 dose of commercial vaccine containing adenoides, milligramides, and galapagos. On day 4, day 9, and day 14, we collected cecal samples from 5 birds from each treatment. Then we extracted DNA. Then we send them out for sequencing using the 16S RNA sequencing targeting the V3 and the V4 region. We used the Illumina NESIC 2000. And then we did the bioinformatics analysis, but I'm not going to go into details of the analysis because of time. I move straight to the results we found in the shift in microbiota composition during Eimeria infection. So we began this from the phylum. At the phylum level, we saw 3 important microbial populations. They are Firmicutes, Proteobacteria, and Actinobacteria. Now we could see that on day 9, which served as a critical period for peak Eimeria cycling, we saw that Firmicutes reduced, then Actinobacteria also reduced in the vaccinated group as compared to the control group. However, as these bacterial populations were reducing, Proteobacteria increased on the 9 vaccinated group as compared to those in the control group. Now, this increment in Proteobacteria could be associated with instability of microbial population at that critical time point. But when we get to day 14, we realize that microbial population begin to stabilize due to partial recovery from Eimeria infection. Now we came down to the family level to look at some important pathogenic family bacteria. Then we saw, again on the 9 vaccinated group, we saw the proliferation of Enterobacteriaceae in the 9 vaccinated group as compared to the control. Then we also saw that the 9 vaccinated group had increased level of Coccidiaceae as compared to the control group. Now, Coccidiaceae could contain potential pathogenic bacteria like Coccidium septicum, which could predispose bird to Coccidia dermatitis, which is a huge disease in the turkey industry. Now, the big question we ask ourselves is that, could Eimeria infection predispose turkey poult to Coccidia dermatitis? That's a question that needs further investigation. Now, McDonald's et al. 2017 also saw a similar thing when they infected Legionella to chickens, and they saw the proliferation of Enterobacteriaceae and Coccidiaceae, similar to what we found in our study. Now, again, we looked at some potential beneficial bacteria at the family level, and we saw the proliferation of Lactobacilliaceae in the 9 vaccinated group compared to the control. Now, this could be due to compensatory response to Eimeria infection. However, whilst Lactobacilliaceae levels increased in the 9 vaccinated group, we also saw the reduction of short-chain fatty acid bacteria like Lactinospiraceae, Rubinococcaceae, Butyriccoccaceae. Now, we all know that short-chain fatty acid bacteria produce butyrate, and they perform functions such as strengthening gut integrity, calming inflammation, improving immunity, blocking pathogens, and improving metabolism. So it means that the reduction in this bacterial population in the 9 vaccinated group compared to the control means that birds on the 9 vaccinated group could be deprived of all these functions, which will negatively impact their performance. Liu et al. 2024 also saw a similar thing when they infected Eimeria maxima to chickens. They saw the proliferation of lactobacilliaceae and reduction in short-chain fatty acid bacteria, which is also similar to what we found in our study. Now we came down to the genus level to look at some potential pathogenic bacteria, and here we also saw the proliferation of Clostridium sensu stricto in the 9 vaccinated group as compared to the control group. Now Enterococcus also increased in the 9 vaccinated group compared to the control. Then Streptococcus also increased in the 14 vaccinated group as compared to the control. Now looking at some potential beneficial bacteria still at the genus level, similar to what we found at the family level, we saw the proliferation of lactobacillus in the 9 vaccinated group as compared to the control. However, all short-chain fatty acid bacteria reduced. Now we look at the alpha diversity matrix using Shannon, COW1, and the observed ASV index. Then consistently in all the index, the matrices that we use, we saw that birds on the 9 vaccinated group, which serve as the peak shedding for Eimeria, had significantly reduced diversity as compared to those in the control group. Now, reduction in diversity simply means functions that are associated with this microbial population will also be reduced. Therefore, the birds on this treatment could be vulnerable to external stresses and other, which could lead to other diseases and other secondary infections. But if we look at those on day 14, we could see that diversity increase, showing recovery from Eimeria infection. But the question we ask ourselves is, could we adapt some strategies or management practices or interventions to maintain diversity at day 9 anytime we vaccinate? Or even if not to maintain diversity, can we beef up diversity a little bit at day 9? And we also ask ourselves that, could we also close down this recovery window from day 14 to maybe day 11 or day 12? We believe that if we are able to do this, we will shorten the instability window and performance loss will also be reduced. We also looked at the beta diversity matrices using Brakettes, Jacquard, and weighted unifrac distance. And similar to what we saw at the alpha diversity, we saw that birds on the 9 vaccinated group showed distinct clustering as compared to the control and also the other treatment. We ask ourselves a similar question. Could we adapt some interventions to be able to close this gap a little bit? And if we do that, we will be able to reduce performance loss. Now, so the summary in the shift in the microbiome composition during Eimeria infection, we saw that cecal microbiota diversity reduced in the 9 vaccinated group. Then we also saw that potential pathogenic bacteria on day 9 and day 14 increased. Then potential beneficial bacteria decreased on day 4 and day 9 vaccinated group compared to their control. Then we saw that short-chain fatty acid bacteria increased in the vaccinated group on day 14, showing recovery from Eimeria infection. Lactobacilliaceae increased in the 9 vaccinated group, showing compensatory response to Eimeria infection. Now, I also want to touch on potential biomarkers for Eimeria infection monitoring. And here, I want to mention that we focused on the 9 vaccinated group against the control because that is where we saw the major shift. We predicted metabolic functional analysis. We saw the downregulation of important energy and amino acid pathways. In summary, we saw that energy metabolism biomarkers such as starch biosynthesis, pyruvate fermentation, arabinose degradation, and gluconeogenesis were all downregulated in the 9 vaccinated group as compared to the control. Then amino acid metabolic pathways such as arginine degradation, valine degradation, isoleucine biosynthesis, phenylalanine biosynthesis were all downregulated in the 9 vaccinated group as compared to the control group. However, as these important metabolic pathways were downregulated in the 9 vaccinated group, we saw that stress adaptive pathways were upregulated, such as glycogen and chitin degradation. So generally, we could see that the microbiome moved from growth supporting metabolism pathways to adaptive response. And we believe that these pathways may be biomarkers we could use to monitor Eimeria infection. Now, looking at the shift we saw in the gut microbiota of turkey poults after vaccination, especially on day 9, and it is clear that the goal is not only to vaccinate, however, it is for us to support birds during peak cycling. Therefore, we recommend that there should be selection of targeted feed additives such as probiotics that can restore short-chain fatty acid. Now, we saw in our study that microbial populations that produce short-chain fatty acids were all downregulated, or they were all reduced. And also energy metabolism pathways were also reduced in the 9 vaccinated group as compared to the control. Therefore, targeted or precise probiotics that could help in the production of short-chain fatty acid should be adapted, so that short-chain fatty acid will be produced to help birds protect gut integrity, then also shorten recovery window. Now, starter diets must include targeted disease interventions to limit opportunistic infection. Now, we saw the proliferation of coccidiaceae, which could contain potential pathogenic bacteria like Clostridium septicum, which could predispose birds to Coccidia dermatitis. Therefore, targeted probiotics that could displace this important bacterial population should be adapted to limit the onset of secondary infection. Now, we also recommend that day 4 to day 9 remains a very critical period anytime we vaccinate. Therefore, special attention should be given to disease prevention at this very critical window to support birds. So for our take-home message, we saw that day 9 is the highest risk period anytime we vaccinate. So it serves as a very critical window of opportunity for timely intervention. We have to plan our support earlier. Cycling is always expected anytime we vaccinate. However, unmanaged disruption increases the risk of secondary enteric challenges and uneven growth. We have to protect gut integrity during peak cycling. Disease management is very critical, especially from day 4 to day 9. We also recommend that there should be the use of targeted feed additives, probiotics, which could restore microbial balance. Targeted short-chain fatty acid production may help mitigate vaccination-related stress and support optimal vaccine responses in turkey poult. Remember, the goal isn't to eliminate cycling, but it is to keep birds feeding, protecting barrier function, and shortening recovery window. I want to thank you for your audience, and I also want to thank my advisor, Dr. Lisa Belke, for her support. I want to thank the Prestige Department of Poultry Science, Avian Turkey Poults, Avian Turkeys, Heavy Farmer, Dr. Kosi's lab, and also members of my lab for the support. Thank you so much for the audience, and I would take your questions. Thank you.
Thank you, Dr. Yeboah.
Okay, so thank you again to our speakers for an excellent and informative presentation. We will now move into the Q&A portion of the webinar. I will begin with the highest rated questions submitted through the Q&A tab. So the first question is, how would you characterize what a healthy gut looks like? Most diverse ratio of specific bacteria species, absence or presence of bacteria.
Right. So this is a very simple question and pretty difficult, right? First indirect answer is that this is not a health. I would not call as a healthy reference, optimal, ideal. I would not call microbiota of broilers or any chickens in commercial production when these chicks are 1, 2, or 3 weeks of age. This is microbiota mostly of environmental origin. Some of them might be chicken adapted, most of them are not. This doesn't mean that the system doesn't work. It works. Chicks can survive, chicks can be produced, and so on. Healthy, I can't tell you whether I'm right, but I really, personally, here I am honest, I trust evolution, right? Because it acted without any human interventions. It carefully selected for the best microbiota of the best composition. So that type of microbiota, which can be found in adult hens, either egg layers or chickens in reproductive flocks, this is something what I would call as a healthy one, characteristic chicken specific, gallus specific. But so this is my answer. And if you want in terms of a complexity, so yes, rather more than less complex, with presence of both gram-negative and gram-positive bacteria, all of this, right? All of this what we observe in adult birds. But there is one critical point when I will now argue against myself a little bit, not completely, right? But keep this in your mind when thinking of microbiota in chickens and associations with commercial production. Evolution selected for a compromise in between of disease resistance, reproduction, body weight increases, all of this. When in broiler production, we can exclude reproduction. If we can somehow exclude infections indirectly via high zoohygienic measures, we do not need to follow exactly what is happening. We do not need to follow microbiota composition, which was selected during the evolution. So I may not be completely right when thinking of specific conditions which are set by human activities. Despite this, so this is what I'm saying you to consider when handling with gut microbiota and when considering what is good one, what is correct one, what is disabled and mistaken one. Myself personally, I do believe that the microbiota, despite all of these critical comments from myself right now, I do believe that the microbiota, which you can find in, for example, in 40 weeks old adult hens, this is the typical and the most appropriate gut microbiota for the chickens. Do not go for exhausted egg layers 70 weeks of age. The microbiota of these chickens is already a little bit corrupted and mistaken one.
Thank you. So the next question is at what embryonoid day was the samples collected and what gut location were samples taken from?
On the corrupted, right, embryonic development, throughout the whole period. So some of the eggs which we sampled were after 5 days of embryonic development. The others were after 18 days. We did not collect that many samples of this type, something like 30, only 30, right? And when we compared microbial composition from day 18 or older stages of embryonic development, corrupted, mistaken one, right? And younger embryonic development, there were no clear associations in terms of microbiota composition. So although probably it's not absolutely correct, 100% correct, I would now answer that doesn't matter. Once the infection of chicken embryos happens, there are no immediate differences whether this happens on day 5 or day 19 of embryonic development. So different ages of the embryos, chicken embryos, different ages of corrupted chicken embryos, no extra consequences for microbiota composition.
Thank you. So the next question that I have is phages are very bacterial strain specific and bacteria quickly builds up resistance against them. The question is this problematic for your solution?
So yes, absolutely correct. There are plenty of reasons why not to use phages, right? And we are entering this area. So I have something like 1 year long experiences in comparison with gut microbiota composition. This is nothing. I admit, or I can say that we failed in any protection of chickens, so really in vivo experiments, when we selected phages against one particular strain. We selected and purified phages through a single pluck. So we really performed the experiment with pure phage cultures, and we then mixed, for example, 3 or 4 different pure phage cultures. So when the mixture phage treatment, when the phage product consisted of 3 or 4 defined, sequenced, and well-characterized phage strains, we never succeeded in decreasing E. coli counts in chicken cecum. So we changed strategy, and this is because of everything, because of the resistances, because of the extreme specificities of the phages and so on. So we changed strategies. We still used a single or a few E. coli strains, but I will explain using a single E. coli strain, and we use experimentally, we use something like 5 different E. coli strains, but I will follow with a single one. And on this single one E. coli strain, which was purified from cecum of 1-day-old chicks sometimes in our laboratory history. So using this single E. coli strain, we infected that one strain with sterile extract obtained from chicken litter from different farms. We infected and lysed and obtained phages, again specific against this one particular strain. When we inoculate these strains or infected these strains with the extracts from multiple eggshell wastes collected during different years, 2 or 3 years in our back in history. So we, in that way, we produced some undefined. In the first instance, we are now going to sequence everything. So, but at the moment we have not sequenced this, so we do not know what and how many phages are present in there. But with that undefined product consisting of, I don't know, unknown number of different phages. So when we use this, we were partially successful. So we then infected newly hatched chicks with our E. coli strain, and we provided mixture of undefined mixtures of multiple phages via drinking water. And in that case, we were successful in decreasing E. coli counts in the cecum, and we could also see reasonably high counts of bacteriophages in chicken cecum. So this is the way we are thinking. If you are familiar with the competitive exclusion product, I think that I can say that this in philosophy, right? In philosophy, undefined mixture of phages. In philosophy, it is similar to the products like competitive exclusion products like Broilact or Avigard, undefined cocktails of bacteria colonizing chicken intestinal tract. So we have not been successful with this. Pure bacterial phages, pure cultures and treatment. We partially succeeded when we used undefined mixture of phages specific to one particular strain of E. coli.
Thank you. So another question, and this one is for Dr. Yeboah. Would early probiotic support help reduce the instability you observed between D4 and D9?
Thank you so much for the question. Yes, I mean, probiotics are very helpful when it's come to modulating the gut microbiome. But the question is, which type of probiotic? I think that is the most critical. So the whole goal of our work was to understand what is happening in the gut microbiome, so that we do not go in for broad spectrum control, but targeted control. Yep. So the question will boil down or depend solely on the type of probiotic that you are using. So in our work, we saw that short-chain fatty acid producing bacteria were reduced. Therefore, if we want to target the production of short-chain fatty acid, we have to go in for targeted or specific probiotics that will help to stabilize or help in production of short-chain fatty acid bacteria. Yeah. So probiotics is very helpful because they have competitive exclusive advantage, which could be used to stabilize the gut microbiome, but it depends on the specific one that we are using. So we have to do very well with our selection to know which type of probiotics that we can use to modulate the gut. It has to do with timing, looking at the window of opportunity. So if we understand the window of opportunity and we select the probiotics very well, they will help. They may not be able to help stabilize it completely, but they would provide, the avenues for the bacterial populations to come back, and to improve diversity. Yeah, but it has to do with the window of opportunity, the timing, and also the specific probiotics that we have to select to target specific restoration. Yeah.
Thank you. We do have another one for you, Dr. Yeboah. Did you use a live non-attenuated vaccine in your research?
We use commercial vaccine for our research. Yes, so it's a live vaccine.
Okay, perfect. Thank you. So as we are coming up to the 1 o'clock hour, I wanted to say thank you again to our speakers and to everybody who has joined us today. We truly appreciate your time and your engagement. Don't forget to visit our websites for more information on these important meetings this summer.